The search for it began 2000 years ago, but it had transformed politics, culture, society, medicine, and technology. And also, it is one of the most powerful, dangerous, ruthless, selfish, and beautiful ideas in the history. This is the story of molecule of life, the DNA, and genes, in which the code of life and our fate is written. January 1933, Adolf Hitler was appointed the Chancellor of Germany. Millions of deaf, blind, mute, disabled, feeble-minded people, homosexuals, dangerous criminals, defective men and women, along with 6 million Jews, 200,000 gypsies, several million Soviet and Polish citizens, were sent to concentration camps and were killed in gas chambers, hundreds of people at a time, using poisonous gases like carbon monoxide and Zyklon B. The victims suffered up to 20 minutes before suffocating to death. Then the bodies were cremated in adjacent incinerator rooms. Bone-crushing machines were used to grind bones. Pile of hair of Jewish men was sent to fabric manufacturing. During the Holocaust, Nazis outrightly murdered millions of people for racial purity of Nazi Germany. They considered Aryans are superior to everyone else. But is it true that a blind man's son is going to be a blind for sure? Absolutely not. Then how exactly traits pass from parents to children? Why do we resemble our parents? And why are we different? The transmission of heredity essentially is the transmission of information. But what is that information made of? In 1850s, an Augustinian monk and a botanist named Gregor Mendel, experimenting on pea plant hybrids, observed that crossing a tall plant with a short one produced only a tall plant, but not an intermediate sized plant. But when he created hybrids of hybrids, shortness reappeared in the third generation. Also, crossing white flowered plants with purple ones, white flowers vanished in the second generation, but reappeared in the third. Traits such as plant height, flower color, and seed texture move from one generation to the next in discrete indivisible particles of information called genes, and genetics is the study of heredity and variation. If genes carry independent traits, then some people thought that desirable traits can be selected and undesirables can be eliminated. What followed was the darkest era in human history, the Nazi Holocaust, selectively eliminating the defective men and women, promoting reproduction of fittest individuals, the eugenics movement. But nobody still knew what genes were made of and where they resided. To understand genes, you have to understand the life of living organisms. All living things are composed of cells, which are building blocks of life. We are made up of 37 trillion cells approximately. By the early 1940s, biochemists had broken cells apart and identified various molecules in the cells, such as sugars, which provide energy, and the fats store energy. And there are proteins, which are made up of amino acids. Proteins are building blocks of body tissue, and they carry out bulk of chemical reactions in the cell. For example, hemoglobin is the protein inside red blood cells that binds oxygen and transports to low oxygen areas in the body. The physical structure of the hemoglobin molecule enables its biological activity. Nearly every cellular function, metabolism, respiration, signaling, growth, cell division, self-defense, and even cellular death requires proteins. Proteins are the workhorses of the biochemical world. But where are genes? Biologists found that genes resided in chromosomes, which are thread-like structures in the nucleus of the cells. Further careful experiments reveal that it is DNA, deoxyribonucleic acid, concentrated like swirling strands in chromosomes, carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. Finally, in the 1950s, using mathematical modeling and 3D images, two scientists, James Watson and Francis Crick, unveiled the structure of the life molecule, the DNA, for which they won the Nobel Prize. It is a double helix with 4 bases on each strand: adenine, guanine, cytosine, and thymine, abbreviated as AGCT. The bases are linked together by the sugar phosphate backbone. Molecular forces between A, T, and GC pairs lock the 2 strands together as in a zipper. A gene is a segment in DNA, a physical and functional unit of heredity. From bacteria to elephants to humans, DNA provides instructions to build an intermediate molecule, RNA. And RNA provides instructions to build proteins. And proteins control all biological processes in the living systems. 3 bases of DNA builds 1 amino acid. ACT, for instance, specifies the amino acid threonine, and CAT, in contrast, specifies a different amino acid, histidine, and so on. There are 20 amino acids after all. A protein is created from amino acids which strung together in a chain. For example, hemoglobin protein, the oxygen carrier in the red blood cells, is made up of chain of amino acids. A small change in DNA can change our fate. In sickle cell anemia patients, for instance, 1 triplet in DNA, GAG, changes to GTG. This results in the substitution of 1 amino acid for another. Glutamate switches to valine. The mutant hemoglobin protein accumulates and changes the shape of the red blood cells like a sickle, which causes excruciating pain in the bones, joints, and the chest, painful swelling of hands and feet, frequent infections, and fatigue. And also, genes build us from scratch. How does an organism arise from a single cell? It is through gene regulation. Few days after the fertilization of the egg by the sperm, embryo forms. A set of genes organize the early embryo into its main axis: head versus tail, front versus back, and left versus right. Then a different set of genes initiate the division of the embryo into its major structural parts: brain, spinal cord, skeleton, skin, guts, and so forth. Finally, organ-building genes authorize the construction of organs, parts, and structures such as limbs, fingers, eyes, kidneys, liver, and lungs. In this process, proteins act as regulatory sensors, like master switches in the circuit boards, turning off and turning on certain genes, or even combination of genes at specific times in a coordinated manner. All cells in our body has the same DNA, but genes express themselves differently in different cells. How birds navigate long distances, for example? Certain genes in birds' eye cells produce a specific protein which help them to see the magnetic field of the earth through which they navigate long distances. After the fertilization of the egg by the sperm, the genes in the father's sperm mix up with the genes in the mother's egg to produce new genes for the next generation by recombination. This is how children are genetically different from parents. If genes are chemicals that produce proteins which control all the biological processes, can we modify genes to produce a particular protein and use it to cure a human disease? Let's take insulin. A protein made of 51 amino acids is released by an organ called pancreas in the human body, which regulates the sugar levels in the blood. In the late 20th century, for the first time in history, a biochemist at a biotechnology company called Genentech separated a gene for insulin from the DNA sequence and inserted it into bacteria, which then produced insulin, which then was used to cure diabetes. Treating diabetes with insulin was a major milestone in the biotechnology industry. Studying human genetics has unveiled hundreds of human genetic diseases. Take Huntington disease, for instance. In Huntington patients, hands and legs move involuntarily with deep cognitive decline and psychiatric disorders. Eventually, patients die of malnourishment, dementia, and infections, yet dancing to the last. Huntington disease is caused by a single mutation in a gene called IT15 in chromosome 4. Another example is Down syndrome, in which children are born with an extra copy of chromosome 21, which results in flattened nasal bridges, wide faces, small chins, and altered folds in the eyes, cognitive deficits, accelerated heart disease, hearing loss, infertility, and an increased risk for blood cancers. Many children die in infancy or childhood, and only few survive to late adulthood. And there is cancer, caused by many genes. Normally, cells grow, divide, and die. All this is controlled by growth controlling genes. But a mutation in these genes results in a cell that do not know when to stop growing, unable to die, leading to cancer, the uncontrolled cell growth. Cancer is caused by the same genes that cause us to grow. Fetuses during pregnancy can be tested for hundreds of genetic diseases, and pregnancy can be terminated if desired. What would you do if the genetic test of the fetus reveals that your future child has 100% risk of Down syndrome? Would you terminate the pregnancy? Genetic tests are also moral tests. The right to be born can be rephrased as right to be born the right kind of genes. Many such abortions are being performed currently. But surprisingly, genes that cause the Huntington disease or bipolar disorder are the same genes that cause our creativity. Sometimes a child with high functioning form of autism performs complex arithmetic calculations, which is the requirement for survival. What if disease causing gene variants are also genius enabling? Mutations are just variations through which life evolves. Genes that adapt better are naturally selected and passed on to future generations. A mutation doesn't imply a disease. The definition of the disease then is a relative mismatch between an organism and its environment. Night on one side of the globe is often a glorious day on the other side. Genes are just chemicals produced by chance of elements on early Earth. These chemicals developed into single cells, which replicated themselves into multicellular organisms such as fish and to ultimately humans. DNA in cell's nucleus is just a molecule that defines us. Human genome contains approximately 3 billion base pairs, which reside in 23 pairs or 46 chromosomes within the nucleus of all our cells. All other apes, including gorillas, chimpanzees, and orangutans, have 24 pairs. It encodes about 20,687 genes in total. In each cell, DNA is about 2 meters long. If you stretch all of your DNA in all of your cells, it would go from Earth to the Moon and back 6,000 times. If we are just genes, then could our genes be modified to treat any human disease, a gene therapy for example, to make ourselves and our genome a little better? Let's take hemophilia, a terrifying bleeding disorder that slows down the blood clotting process. In normal people, F8 and F9 genes in the liver cells produce particular proteins called coagulation factors or clotting factors, which release into the blood and combine with platelets to clot the blood after an injury. But mutations in these genes produce abnormal proteins, which cannot clot the blood effectively, leading to excessive blood loss. In 2014, a successful gene therapy was announced to treat hemophilia. Scientists modified a gene for clotting factor and placed it into a retrovirus and infused the virus into the patient's body, which dramatically reduced bleeding. But this does not affect more than one generation, as the changes do not affect the reproductive cells, that is sperm cells or egg cells. To create permanently modified human genome, changes have to alter the genome of the reproductive cells to pass on to the next generation. But change in the genome of the reproductive cells currently is illegal because of ethical challenges. Future is exciting but also concerning. Gene therapy or genetic engineering might make us immortal but also mortal. What if a genetic experiment goes wrong and triggers a deadly virus that causes our own extinction? An intelligent organism has learned to read and write its own instructions, but it is such capacity that empowers us but also makes us vulnerable. But by what standards we call ourselves intelligent? How about some life elsewhere in the universe that is based out of completely different molecules and genes that are a million times intelligent than us? After all, it is the genes that want to survive, through an experiment which on earth we call life.
Me to feel I'm falling.